All right, welcome back, everyone. So we're going to pick up here and continue talking about our immune system, specifically in the innate immune system. We're also going to stay discussing kind of that second line of defense, right? So we're called the first line of defense. That was our barriers, right? So think skin, mucous membranes. Second line were our cells and various cellular processes, right? And again, for all of these, remember, the innate, these are non-specific, right? So remember, these are our non-specific defenses, right? So what we're going to look at now are a few internal cellular processes, and some not even really involving cells, that are going to take place that act to eliminate some type of pathogen, right? So these are going to be various types of antimicrobial proteins that we'll talk about. We'll talk about something called the complement system. And then we're going to talk about something called interferons, right? Interferons. So we'll start here by talking about interferons, right? So interferons are these, essentially think of them as antiviral proteins, right? These antiviral proteins or these antiviral molecules that are released from a virus-infected cell, right? So the cell is already infected with a virus. At that point, the immune system in our body essentially considers that virus-infected cell it's a goner, right? It's going to be sacrificed, right? It is no longer viable. We're not going to try to save it. But what we are going to try to do is ultimately save the other cells, right, that are not yet infected with the virus, and hopefully prevent them from being infected with the virus, right? So what you have initially happen are the virus-infected cell begin to release something called interferon alpha and beta, right? And so these are produced by leukocytes, and these are also produced by virus-infected cells, right? These interferons, the interferon alpha and beta, these will then bind onto neighboring cells. So they're acting in kind of a paracrine signaling fashion, right? So paracrine meaning they're acting on the cells kind of nearby, as opposed to endocrine and autocrine, right? And they're going to stimulate those non-infected normal cells, right, to produce antiviral properties, antiviral proteins, but also produce enzymes that destroy viral nucleic acids, right, and prevent the synthesis of new viral proteins, right? So if they were to become infected, they're going to prevent the synthesis of the information coming from the virus, right? So in that case, the viral nucleic acids stop the production of viral proteins, right? So this is how viruses propagate, right? This is how they act, right? You have to have a host in order for the virus to continue, right? So whether you're talking about the flu virus, or you're talking about the measles virus, or you're talking about COVID-19, right? Whatever you're talking about, that virus has to have a host, right? So in that case, viruses are typically considered non-living, right? They're not alive. They have to have a host in order to survive, right? So interferon beta, right? They can also stimulate natural killer cells to destroy infected cells, right? So through those perforin and granzymes that we talked about earlier. And then some of them can also stimulate the release of interferon gamma from T cells and natural killer cells. And these are going to stimulate macrophages, right? And these interferon gamma molecules will stimulate macrophages that then go and endocytose and engulf and destroy cells that are already infected, right? So the big idea here is that you're not necessarily trying to save the infected cell, but you're trying to save cells that are not yet infected from becoming infected, right? And so this is this kind of interferon process, right? The second one is called the complement system, right? So the complement system is it's a group of proteins that are in the blood, and they're going to work together or complement each other and complement antibodies that are produced by cells called plasma cells that we'll talk about later, right? So these plasma proteins are produced in an inactive form, and they're systematically going to become activated. And there's kind of two main pathways for these proteins to become activated, right? One is called the classical pathway and one is called the alternative pathway. The classical pathway involves antibodies attaching to a foreign substance, okay? So meaning some other pathogen, right? Whether that be a virus or a bacteria or something that is non-self, right? It's abnormal, it's foreign. And then these complement proteins are then going to bind to the antibodies, right? The alternative pathway is that the complement binds to the walls of fungal cells or potentially bacterial cells, right? And so when you have the complement system activated, and I'll show you some images in a little bit, you have a few processes that result, right? So one of these are inflammation, right? So the complement system enhances and stimulates inflammation, right? It's going to also activate or result in something called opsonization. And so this is essentially when you have antibodies and some of these complement proteins essentially kind of cover a pathogen, and as a result of covering the pathogen, they increase the likelihood that that pathogenic cell, that kind of tagged pathogenic cell, is going to be phagocytosed, right? So it's essentially tagging cells to then be eliminated. And then the big thing is something called cytolysis, right? Which is essentially cell rupturing, right? And so or one of the things that this complement system produces is something called the MAC, or the membrane attack complex, right? And so this membrane attack complex is essentially going to puncture a hole in the cell wall of a pathogen, so maybe the kind of outside of a virus or the cell wall of a bacterium, and this is then going to allow massive amounts of fluid to kind of pour into the cell and then ultimately cause it to rupture, right? So it's going to trigger it to be destroyed, right? So this here is kind of a overview of some of the effects of the complement system, right? So you can see increases inflammation, eliminates immune complexes, so with an antibody and an antigen that we'll talk about later, cytolysis that we just mentioned with the MAC, the membrane attack complex, and then things like opsonization, right? So kind of covering the pathogen to make it more visible, right? And so this whole complement system, you can kind of see in the middle here, it's a network, it's a system of 20 or so proteins that are circulating in the blood. They'll become activated. So almost think of this as kind of like how our clotting cascade with blood, the various clotting factors systematically become activated. So too these proteins are systematically becoming activated. And then the end result is to ultimately form that MAC complex, right? That membrane attack complex, right? So here you can see a few additional things. On the left you see the sequence of events in the classical and the alternative pathway, right? So classical pathway involving the binding of antigens or antibodies to an antigen, and then the complement proteins binding to that. And then you have down here this kind of systematic cleaving and activation of one complement protein to the next, right? With the end result being this membrane attack complex, and you can see that here, that are inserted into the wall of a cell, and then that allows extracellular fluid to rush into the cell, stimulating it to ultimately lyse, right? Stimulating it to ultimately burst. Okay. So a few additional things that we can talk about here with relation to our innate immune system. One of these is inflammation, and so we've all, you've likely heard of inflammation, right? Inflammation, think of it as this kind of localized swelling in certain areas. And what this is doing is it's acting to kind of block off unwanted substances from reaching the tissue area, but it's also helping to bring helpful cells and helpful chemicals and other things to the site of infection, right? So this is going to involve our tissue, basophils, mast cells, and the infectious organism that's going to release various chemicals, right? So some of these chemicals are histamine, what are called leukotrienes, prostaglandins, and other types of chemical chemicals from various cells, right? So when you have inflammation, right, again, you have major vascular changes, right? So imagine changes in the size and permeability of our blood vessels, right? So main thing is vasodilation, but also increased capillary permeability, right? So the vasodilation is helping to bring additional blood and immune cells to the site of injury. But then the increased capillary permeability is helping to allow some of those immune system cells to then leave the vascular system, go into the tissue where the actual infection is taking place, right? So there's a couple of things that are involving or that are used to recruit these white blood cells, right? And so these are processes called margination, diapedesis, and chemotaxis, right? And I'll show you an image here in a little bit that kind of walks you through some of those, right? And so I'll leave that for discussion here in just a second, right? You also have plasma proteins to the site of injury, right? So these include immunoglobulins, so antibodies, right? Some of the complement systems, clotting proteins, kinins as well, that act to help with essentially allowing the white blood cells to kind of adhere to our blood vessels, right? And so we'll talk about some of those here in just a little bit, right? You also develop exudate, so this fluid that's moving from the blood to the injured site, right? So to the infection site. You're also losing plasma proteins that result in a decreased capillary osmotic pressure, so less fluid remaining in the blood, so additional blood in the tissue, right? So again, contributing to the swelling that you associate with inflammation. And then you'll have things like increased macrophage activity, tissue repair, things like that with some type of tissue injury, right? So this here is showing some of those kind of main processes, right, involved in inflammation. So you have some type of tissue injury, right? So you scrape your knee, you cut your finger, something like that, right? Bacteria enters, right? So from the external environment. You have the release of these chemicals, so think histamine, heparin, things like that. And then you have the activity of your white blood cells, right? The white blood cells. So they begin moving, they begin undergoing this process called margination, right? And so if you look back here where I talked about it, these three here, margination, diapedesis, and chemotaxis, the white blood cells initially move to the outer side of the blood vessel, so to the margin, hence the term margination. Diapedesis, they begin squeezing through the blood vessels, so squeeze through the cells of the blood vessel and leave the blood and enter the tissue. And then chemotaxis, meaning the cells are attracted and moved toward the actual site of the injury, right? So neutrophils, things like that, right? And so you can see some of those things happening here, right? They undergo margination, they move to the side. Diapedesis, they squeeze through. And then chemotaxis, they are attracted to the actual site of the injury. Okay. So whenever you have this process occur, when you have tissue inflammation, right, there are several kind of cardinal signs or things that you can recognize that are associated with inflammation, right? So these are things like redness, heat, swelling, pain, and loss of function, right? So redness is sometimes called rubor, swelling is called tumor, heat is called calor, and pain is called dolor, right? So acute inflammation is normal, right? So due to some type of tissue injury, something like that. Chronic inflammation can have detrimental effects. You can end up with loss of overall mobility. You can have the replacement of normal healthy tissue with more fibrotic tissue, right? So collagen fibers that are replacing normal healthy tissue. So you can have negative consequences to long-term chronic infection. But acute infection, again, as a result of tissue injury, things like that, that's normal, right? So you can think of the cardinal signs of inflammation kind of as a graphic like this. So again, you've got heat, you've got redness, swelling, pain, and then this loss of function, right? And so again, think of any time you've sprained an ankle, you have accidentally cut your skin with something, or you had a scraped knee or something like that, right? Think of something that would cause inflammation, and think about some of these various signs, or these cardinal signs that you would expect to see with the inflammation. Okay. So last kind of main thing we can talk about here is fever, right? So fever is a major part of our innate immune system. So again, non-specific. And so fever is an elevation in body temperature, right? So let me just say one thing. Whenever you have a fever, right, or when you are referring to somebody who has an elevated body temperature, don't say, oh, they have a temperature, right? Oh, they're sick, they have a temperature, right? Well, everybody has a temperature, right? Everybody has a normal body temperature. What you're trying to say is you have an elevated body temperature, right? An abnormal body temperature elevation, 1 degree Celsius or more from normal, right? So say normal is something like 98.6, you're 1 degree or so, 1 degree Celsius, that's Fahrenheit, but 1 degree Celsius or more above that normal, right? And so a fever results from the release of something called pyrogens, right? And pyrogens are simply cellular chemicals that result in the increased internal body temperature as a result of essentially resetting our internal thermostat, right? So our internal thermostat is in our hypothalamus, right? And so you can read through some of the events of fever. Typically, they're described as being the onset, the stadium phase, and then something called defervescence, right? So the onset is the onset of the elevated temperature, so the onset of the fever. Hypothalamus resets our internal body temperature. You get constriction of blood vessels. So we're trying to reduce the amount of heat that's lost, so we retain more of the heat, so you end up having an increase in body temperature, and then you may shiver as well, right? So muscles generating more heat. You have something called the stadium phase, which is where the temperature, the elevated temperature of the fever is more or less maintained, right? It's kind of held at a constant state. And then you have the defervescence phase, which involves the temperature breaking, going back down. Our hypothalamus no longer responds to these pyrogens, or they're no longer present, and the body temperature decreases, right? And so we return back to normal, right? So this is showing some of the benefits of fever, right? And so fever is a normal thing, right? It's normal if it is healthy, it is purposeful, right? It promotes interferon activity. So remember, interferons have to do with our viruses, right? So antiviral activity increases the effectiveness of our adaptive immunity that we'll have a separate discussion on. Accelerates tissue repair, increases CAMs. CAMs, these are cell adhesion molecules on the endothelial cells in our capillaries that allow for those leukocytes to leave the blood, so that diapedesis, and migrate to the tissue site or the infection site. And so again, fever itself is normal, right? And so again, you're talking about gradations of fever. Okay, so low grade 100 to 101 degree, intermediate 102 degree, high grade 103 to 104 degrees Fahrenheit, and then dangerously high is anything over 104, right? And so the reason for this is that you can end up, you can actually cause something called febrile seizures, in which the elevated body temperature damages the brain and results in seizure activity. You can also damage the brain tissue itself. You can end up with denaturing of proteins, right? So remember, one thing that affects protein function is chemicals and temperature, right? So you can denature those if the internal body temperature is too high, right? And so a low-grade fever, again, it's considered normal. It is typically allowed to persist, but if it continues to elevate, again, 104 and above, that's when it becomes potentially very dangerous, right? Very dangerous, right? And so we can talk about inflammation. I probably should have had these slides a little bit earlier when we were talking about inflammation. But talking about acute and chronic inflammation, right? And so you've probably all heard of the Rice method, right? So rest, ice, compression, and elevation, right? So applying ice can help with acute inflammation. So vasoconstriction of blood vessels help decrease the inflammatory response, but it may interfere with the healing process, right? So again, allowing the blood to flow to the area, allowing white blood cells to leave the blood and enter the tissue site, right? If you are constricting the blood vessels and reducing the flow, then you may hinder some of that healing process, right? But one thing it also is good for is ice. The ice it is to help with pain, right? So we all know that low temperatures can almost act as an anesthetic, right? And so that's one potential benefit for ice in periods of acute inflammation, is not only to help reduce the swelling, but also assist with the pain as well, right? Now, if you have chronic inflammation, this is inflammation occurring for longer than 2 weeks. Instead of the neutrophils, which are more associated with acute inflammation, you have macrophages and lymphocytes. This can result in more potentially irreversible damage, so tissue destruction, developing scar tissue. This could be due to an autoimmune disorder, right? So maybe something like rheumatoid arthritis, something along those lines. This can also occur due to overuse, right? So tennis elbow, shin splints, osteoarthritis, other things in which you are putting repeated stress on the body, in which you develop inflammation, but then the inflammation just persists because of your continued activity, right? So again, whether that's running, whether that's some type of sport, right? You kind of keep that stimulus present, and so the inflammation stays, right? It is continuous, right? And so there are various medications you can take, right? So you've heard of NSAIDs, so non-steroidal anti-inflammatory drugs. So think Advil, right, is a good one, Aleve, things like that. So these are good at reducing acute inflammation, helping with pain as well, but they're not without their side effects, right? So Advil, things like that, are typically they can be damaging to the kidneys. They can also result in gastric irritation as well, especially Advil, right? You can end up with gastric ulcers if you take them for prolonged periods of time. Okay, so here are some questions to consider, and this is where we will stop for our innate immune system, right? Remember innate, think non-specific. It's more immediate, right? And what we'll begin talking about in kind of the next set of lectures is the adaptive immune system, in which we will focus primarily on our lymphocytes, so our T cells and our B cells, and we'll talk about something called cell-mediated immunity and something called humoral immunity.